All right. Oh, okay. Great. Well, welcome everybody to today's QLS seminar. It's my great pleasure to introduce Professor Colijn. And this is a lot of fun for me because I work with Colijn on a daily basis as part of COVARNet, the COVID Variants Rapid Response Network. But it's actually rare that I get to hear about some of the science that she's doing in more detail. So this is a particular pleasure for me, and I hope it is for you too. So Colijn did sort of a tour of Canada and the US and the UK and back in terms of her academic trajectory. She did her undergrad at UBC, followed by a master's at York University in Toronto, and a PhD at the University of Waterloo. She then did a postdoc at McGill with Michael Mackey, and a second postdoc fellowship with Megan Murray at the Harvard School of Public Health, where we actually first met and collaborated on a few projects on genomic epidemiology and bacterial GWAS studies. She then took a first faculty position at the University of Bristol in the UK, and then moved to Imperial College London, where she stayed until I think 2017, 18, before moving to Simon Fraser University. So she came all the way back to Vancouver, and we're thrilled to have her back in Canada as a Canada 150 research chair. And as she'll tell you all about, Colijn is a epidemiologist and mathematical modeler interested in also various different aspects of infectious disease biology. So again, thanks, Colijn, for joining us, and looking forward to your talk, and welcome everybody.
Thank you so much. Thanks for the really kind introduction, Jesse. It's nice to see you and Carmen Leah here, familiar faces from weekly and more often COVID meetings. So yeah, great to be here. I'm going to share screen. And hopefully this is now in a full screen mode that is appropriate. So I'll start with, I wasn't sure exactly what the background was and if this is pitched right for this audience. So please, if something's not clear, just jump in and ask a question, because it's better for it to be clear for everyone, hopefully, than or for than for people to be wondering. So I'm going to talk about genomic epidemiology in SARS-CoV-2, some of the new work we're doing, and then some remaining challenges that I think are preventing this area from really making the progress that it could be making and offering us what it could be offering. So I'll start by saying something about what is genomic epidemiology. It's a phrase you're likely to hear, and I would broadly describe it as the use of pathogen sequences, so RNA sequences or DNA sequences, to help us understand transmission or answer epidemiological questions. And here, transmission can mean lots of things. It can mean global geographic movement of the virus, which we try to understand using sequences. It can mean past population dynamics of the virus. Has the population been larger in the past and then gone down and gone up again? Or it can really mean drilling into that person-to-person level at a high resolution, kind of really who infected whom or who didn't infect whom, and what does that mean in terms of transmission or in terms of public health interventions. And the reason this field is exciting and new in part is because sequence data, the RNA or DNA of the pathogens, can offer this very high resolution view of how pathogens spread. The resolution's not perfect, and the technology is pretty new. So since about 2007, sequencing technologies became vastly, vastly cheaper, even compared to Moore's law. There's a plot you may have seen. You have sort of a log scale, and Moore's law is, things getting, computers and so on getting cheaper by a factor of 2 every 18 months. And sequencing just plummeted, even compared to that, got sort of 5 orders of magnitude cheaper. And that gives us the opportunity to look at infections in this really high resolution way and try to understand how they're spreading and use that information to do something. It's kind of a new field. It's established a lot of tools, but in terms of actionable things, it's maybe still missing some methods. Okay, so this just kind of illustrates the idea. You have two sequences. They have A's, C's, T's, and G's in them. These two differ at 2 points, here and here. And imagine we have the whole genome, so not just these characters, but many thousands or millions of them. We can use these to estimate how closely related two people's infections are. If they're super, super closely related, that suggests recent or maybe direct transmission. If they're really distant, that indicates they did not infect each other. And so the challenge is to use that heuristic to make inferences about transmission that are specific to locations or to lineages of the pathogen or to small clusters or groups of people, and ideally to do that quickly and quickly enough that it's actionable. Okay, so this is one schematic view of what one of the aims is in my research group. These kinds of trees, you've seen them, Carmen makes them all the time. The dots indicate people's virus sequences, and the nodes inside the tree reflect patterns of ancestry and relatedness. So it's a phylogenetic tree. So we could identify clusters or groups of sequences, try to understand them in more detail about who might have infected whom, and analyze their dynamics, and then use that information in modeling to try to think about what those virus sequences mean about what's happening with transmission. Okay, so in particular, when we say modeling and what does something mean about transmission, what I'm going to talk about here is a method to estimate serial intervals. And what are they and why do we care about them? So the serial interval is defined as the time between symptoms in a person A and symptoms in the person that A infects. So infector A, infectee, how long between symptoms? Of course, in COVID-19, not everyone has symptoms, but everyone in our data that we have a sequence for has a collection date when their sample got collected. So they had to be tested, they got tested. And so we use collection date when we can't access symptom data. There's a lot you can say about serial intervals. And if you're one of the students who read the papers that I circulated before, there are various methods to estimate these, but all of them need a certain basic piece, right? If you want to think about the time between symptoms in A and symptoms in someone that A infected B, you need to have some pairs where you think A infected B. If that happened, okay, serial intervals require these data, who infected whom, and they have a lot of kind of onward implications because it helps us understand how quickly an infection spreads, and they also help us understand how much transmission happens before symptoms. And that's not just from serial intervals, where everyone has symptoms, that's from comparing the time between symptoms in A and B to the time that A got infected and then when A got symptoms. So you need these sort of two things, the symptom one and the incubation period, infection to symptoms. I won't go into that a lot. Serial intervals are also fundamental to estimating R0, the basic reproduction number. And that in turn is the kind of key basic fundamental parameter of all of the infectious disease modeling, including estimates for the portion of the population we need to vaccinate. All those R sub T estimates you see on Twitter from David Fissman and otherwise, is it 1.2, is it 1.08, is it 2? Those all rely on serial intervals. That's all underneath there. Because if you want to think about how many cases on average a case is causing, and you look at case count data, these methods go back in time and say, okay, so 5 days ago there were 100, and then 4 days ago there were 102, and then you kind of integrate over this serial interval distribution to figure out how many new cases there are. So my point is, these require A infected B information, and they are fundamental to lots of modeling efforts that happen after they are estimated. For COVID, the world is still using serial interval estimates from Tianjin, China, and Singapore from early 2020, because there's so little data available. And that's for two reasons. One is it's hard to get these data where you really think you know who infected whom. And another is if you ever do get it, you tend not to share it because these are gathered by public health and they're individual level data about who went to which shopping center and which church on which day. Singapore shared a lot of this data that we would never in Canada be able to share. And in general, they're not shareable data. Okay, so we wrote a paper. The first paper we wrote in the pandemic was in early 2020. And we looked at these data from Tianjin and Singapore, and this is very detailed contact tracing data, but even so, many people attended the same church or shopping center or event, and it's a bit hard to unpack exactly how the transmission routes might have happened. But once you do, you find these pairs where you think A infected B, and then you go, okay, how long did it take between their symptoms, infectee, infector, and you go, all right, like here are some days. And then you do statistics that take into account that we only see these pairs when B has already occurred. So if you're sitting here on this date on February 20th, and this person infected someone and you haven't seen them yet, your data don't have that. So you have this right truncation. And this, I've shown the bottom part of this picture. It actually goes up diagonally in order of the infector timing. And so you do hit up against this wall where it was the present day at the time we were doing this. Okay, and you can try to account for some missing information that way, the people you haven't seen yet, and maybe people who occurred in between, so we don't really know that A infected B. Okay, so that's kind of a quick guide to how you estimate this fundamental parameter in a conventional way without sequences. What we want to do is use virus sequences kind of as a proxy for that contact data. And that's an opportunity because sequencing of viruses is now done routinely in many places for other reasons, to monitor public health, to look at outbreaks, to look at variants. And that gives us an opportunity to try to understand this fundamental modeling parameter using a new kind of data. And so the idea is, if A and B have viruses that are really closely related and A and B occur in the same place with realistic timing, then A and B are a plausible transmission pair. Doesn't mean A infected B, but they're plausible. If A was observed first or had symptoms first, A might have infected B. That's the limitation of this model. We don't do the negative serial intervals, but that allows us to connect virus sequence data and serial interval estimates. So I'll talk a little bit about the data that I'm going to show. This is in collaboration with a group at the University of Melbourne and with Melbourne Public Health, I guess led by Ben Howden, but our key collaborators are Courtney Lane and Anne Watt. We have data from two so-called waves of SARS-CoV-2 of COVID in Melbourne, the first one and the second one earlier in 2020 and later in 2020. And so this just shows the cluster sizes. The top where the line goes is how many people they ever had and the kind of dynamics of the clusters over time. And you see in wave 2, we have some bigger clusters. These are defined in slightly different ways, these clusters. And I've just illustrated for wave 1, the tips on a phylogeny. So this is one of these trees where each tip corresponds to somebody's virus. And the internal nodes of this tree reflect the relatedness patterns. So this crowd of gray folks here would be their viruses are descended from the virus on this branch here. And we've colored in the clusters that, that we analyzed for the wave 1 data. Okay, so in brief, the approach is to start with these clusters, same picture, same tree. And these are based on the virus sequences, how close they are. You can see closely related viruses are grouped together in the same color, which is the same cluster. They're also based, especially in wave 2, on some of the epidemiological data. So were they at the same place around the same time? We identify plausible transmission pairs using genetic distance and differences in timing. Then we sample plausible transmission trees that are composed of these plausible transmission pairs. For each of those trees, then those trees are now who infected whom. So we have infector and infectee, and we have a whole story of the outbreak of the cluster, but we don't know it uniquely, so we have lots of them. We use them to estimate the serial intervals, and then we combine the estimates to capture the uncertainty. Okay. And here is a schematic of the whole pipeline. And it illustrates some of the parts that I haven't talked about. I'm not going to talk about the sequencing technology and the strategy and the bioinformatics that go behind, that goes on behind getting these sequences and computing their distances, but that all happens. So some virus is taken out of someone's nose, it tests positive on a PCR, and then sequencing happens and you get a consensus sequence for each individual. That's the ACTG string. Then we make a tree of the kind that you just saw with the dots and the colors. And we look at genetic distances between the individuals and combine that in some cases with exposure sites. That gives us a transmission cloud of these feasible pairs. And we get these transmission trees combining these sort of plausible pairs. And then we use a statistical model that I'm about to tell you about to estimate the serial intervals. Okay, so here's the statistical model that I'm now going to tell you about. It has three different parts. So the main thing we want to estimate is the time between two people, right? A and B, A infects B. When did they get symptoms? How long did it take? And that's not always going to be 5 days. It's going to be 5 days plus or minus some days. So we model that with a gamma distribution. It has a shape and a scale parameter. Then we think, well, okay, we never actually know really who infected whom. And especially with our virus sequences, we're just not going to know this. There might have been some people that we missed in between. And the number we missed is going to be related to how much sampling we're doing. We might know that we're really making an effort to sequence every single case, or we might know that, we're kind of getting 30% of them. So we have some unknown number of gray individuals here who are not sampled. That's geometrically distributed, the number of them. And that gives us what's called a compound gamma, geometric gamma distribution for the time between red symptoms and other red symptoms, the two people we do have. And then the other option is, they might not have infected each other. They might both have been infected by an unknown person. So this co-primary transmission. So we have some fraction. The likelihoods of these things depend on how many people we're sampling, and they impact the timing between the two individuals. So here we would have a gamma difference distribution for the time between observed symptoms in red here and the other red here. Okay, so in the end, that gives us a statistical model that has 4 parameters, and it models the time between the symptoms or the collection date of I and J being the 2 red individuals who are in our sample. And that's if they have a, this is 1 minus W. So this is the gamma difference distribution. So this is from the last term here. And then if they are not co-primary, so these other roots, then you have this compound gamma, compound geometric gamma distribution here. So we want to estimate 4 parameters: the essentially shape and scale, or mean and standard deviation of the underlying distribution we actually want, and then these 2 other parameters: what's our sampling fraction, and what's the portion of our pairs that have this phenomenon happening where an unknown individual infected both the people. Okay. So of course, we have these plausible pairs. We're defining as 2 people who are close enough in symptom onset time or collection date, and whose viruses are close enough genetically. So we just say, you know what, 30 days out, you're not a direct link anymore. You're not even plausible in our model. We have to just make a cutoff. And similarly, a genetic distance of, say, somewhere between 1 and 2 mutations in the virus, as measured in the genetic distance between the 2 viruses, it is going to be too much, or maybe it's 3 or 4. We have to pick a cutoff where we say, okay, that's too much, but then within that, we allow these indirect routes. So in doing that, of course, cases can have multiple possible infectors. You just have a sequence, and you have all these people who are nearby with time and nearby in genetic distance. So our approach is to have each individual has 1 possible infector at most in the data set. And we select that infector with a probability that is higher for a more closely related virus. And it's higher for a nearby time. So that means if 2 individuals have virus sequences who are really similar and they're 2 days apart, the probability of a direct infection in our model is higher than if they're 30 days apart and they have more distantly related viruses. Okay, so we do that all probabilistically, and we sample lots and lots and lots of plausible transmission trees, kind of like this, except this isn't a tree. And then we use them all because they're all plausible. Here are a couple of sampled trees and graphs from the Wave 1 data from Melbourne. You can see you get individuals with nodes and you get edges between them in the sampled tree, and these little gray numbers show how many days between between them there were. And I want to remind you that 8 days or 14 days are not really that plausible for COVID for direct transmission, but this adapts for the fact that there might have been unknown intermediates in here. We don't have every case in our data. And then we can visualize these with a weighted graph where we put a whole pool of these transmission trees and then say, okay, well, look, this person was probable that they're the infector of this person under this model, and so on. And these trees themselves are potentially interesting for public health, just because they can illustrate what the data, when synthesized in this way, can tell you about who infected whom. Okay, so then what we do is we maximize the likelihood of our statistical model, having identified all these edges where we have A infected B and it took them 3 days, but we have this model where that 3 days comes from these different routes. And what we find is serial intervals that are like pretty consistent with standard estimates of around 5 days, pretty variable. They differ from cluster to cluster, but if you actually go like, hey, is this 3 like significantly different from this orange? Like, no, it's not that different. Okay, but that's nice, sort of a sanity check. You get a reasonable number for the mean, you get some reasonable and fairly wide number for the standard deviation, and you say like, okay, that's nice. We can drill in a little bit further. These are the second wave Melbourne data, and here I should note that the clusters, the groups that we put in are defined a little differently because if you have lots and lots of transmission locally, and you try to sequence everything, then, and you have a pathogen that goes from person to person, there isn't really a meaningful division into, you know, neat clusters that are separate from each other. You get A is linked to B and B to C and C to D and D to E. And of course it goes on, and pretty soon you have 8,000 cases. So public health really loves to kind of cluster their sequences into little groups and then explore the little groups, but that's not always meaningful in this kind of data. But here, so these are some exposure sites, as well as using the virus sequences to help define the groups. Then here are the groups. And now we do actually find some differences that are maybe a bit more pronounced between some with shorter and some with longer serial intervals and some with lower and higher standard deviation of the serial intervals. Okay, so what does that mean? I started out by motivating this, talking about how important serial intervals are because they impact all this other stuff, right? They impact all this modeling and RT. And indeed, one of the motivations for this is to try to use sequences to reveal differences in transmission dynamics. Some of those might be due to the virus, or they might be due to local epidemiology. What's the context? What's the population in which the virus is spreading? So here we use these red blobs to illustrate, you know, an index case and their infectivity over time, and to explore questions about what's the cost effectiveness of rapid testing, how quickly people are infectious and infect each other impacts how soon you have to find cases before you can stop transmission chains. So it impacts these practical questions, and there's room for a lot of heterogeneity. In the Melbourne data, we found, this picture appears just fine on my computer, but it does not appear here, which is odd. Let me see if I escape this, if it comes up. No, that is very strange. Okay, I'm going to check the time. What time is it? All right. Okay, what do I do here? Let me just see if I may not be able to find the appropriate tab in which that picture appears without, maybe I will. One sec, let me see if I can find the tab where that's there. Yes, okay. These, so I'll talk about what this is first, and then I'll show it to you. And I'll briefly just stay out of the presentation mode. Okay, so effective reproduction number is the average number of new cases that a case causes over their infectious period. If this number is bigger than 1, each case is causing more than 1 case on average. If it is less than 1, the cases are declining, and are not, if there's no immunity and so on, impacts how many people have to be vaccinated to get sufficient immunity for cases to decline. All of these things depend on the serial interval. So we can ask, hey, we got these different serial intervals. Does that actually mean anything about transmission when seen through this lens of reproduction numbers? And the answer is sometimes. So these are the wave 1 and wave 2 data from the paper, and I'll make it bigger so you can see it. And it shows that each color is a cluster, each little panel is a cluster, and it shows 2 different estimates of the RT over time. They all go down because you always end the cluster, so they go down. I think the important thing is what they look like at the beginning. And mostly our estimates and the standard estimates are the same, so these things overlap. But sometimes they're quite different, at least at the beginning, like in this one. The fact that we got a different serial interval for this cluster actually means we would estimate its effective reproduction number at the beginning as only between 1 and 2, and not between 3 and 4. So it's a really different pattern. And you see some of those differences. Mostly they're the same, which is kind of reassuring, but sometimes they're quite different. Okay. We can also think about the transmission setting, and this is the wave 2 data from Melbourne. The mean serial interval is shown here in our estimate, and the color indicates the setting, the transmission setting. Again, I wouldn't stake okay, there's a significant difference between all the meat packing and all the healthcare, for example. But we did see more of the orange meat packing processing at the bottom end of this, smaller serial intervals. And we saw more of the healthcare facilities and housing that tended to be on the upper end. It's not clear why, and it's likely that this is not significant. You could probably slice and dice this, if you looked at occurrence of these in the lower ranks and occurrence in the higher ranks, you could probably find something. But I would say this is sort of suggestive and interesting in that setting did seem to make a difference here with some of these longer ones in healthcare and housing and some of the shorter ones really in meatpacking, where we saw very, very steep initial rises in COVID transmission. Okay, there's that one where it did actually come through. All right, that's what happened there. I'm going to talk about 2 comparisons because this is a new method, and it's sort of, I don't know if I've conveyed the level of skepticism that people who are more used to the conventional methods might exhibit about, how could you possibly use these virus sequences? So many of them are so similar. So we want to compare our estimates in 2 ways to what you would get from standard methods. So here's one where we didn't use genomic data to define plausible pairs. We used contact tracing data, which Public Health in Melbourne shared with us very generously. And we found quite similar estimates for the clusters, same kind of range, same kind of smallest ones around 3, largest ones around 8 or 9, and really high variability. This is just for wave 1, where they had more capacity to do really detailed contact tracing data before they had lots and lots of COVID in Melbourne. So this data are not available everywhere. And of course, the people in the clusters differ a little bit because if you define the clusters differently, so they're not comparable in terms of like cluster 13 is the same group and we can't really directly compare, but we do get fairly similar estimates. So that's using the contact data instead of the genomics, but then using our same statistical model, imagining, okay, we might have missed people in between, or we might have missed somebody who infected both the red people. So that I think causes this high variability. So we also compared our estimates to previous estimates from other populations. You can see Singapore and Tianjin, and now there are a few more, not all publicly available data, but the estimates are available. And these use contact tracing data from your more standard intensive epidemiological investigation. So they might have better transmission pairs, right? They actually asked people, where were you? And was this your husband or this your best friend who you were in contact with? And how long was the contact? All we have is these virus genomes that are, we don't really know as much about the pairs. So these previous estimates tend to be narrower in their estimate of the mean serial interval, but they also don't account for these unknowns. They tend never to account for this idea that if you see 2 days in between, it might have actually been a shared exposure to someone else 3 days and 5 days prior to that. So your interval might be 3 or 5, but you're seeing these 2-day differences because of that co-primary root. So that's another reason why published estimates might be much tighter than our estimates. They tend to assume sampling is perfect, and so they don't need to account for, were you both infected by the same additional person we didn't see. I would say both the mean and the variability here are really important for public health measures. Mean is probably most important in terms of this, how many people do you need to vaccinate, because it really feeds into those estimates of R and the numbers of new infections. But the variability is important too. It will feed into those onward transmission modeling estimates, but it will also feed into really practical questions around isolation time and testing frequency. Okay, so what's next? I think I'm probably half an hour in-ish, yes. I want to talk a little bit about evolution. This is for Carmen and Jesse. This is an old version. It doesn't have Omicron on it yet, so it's quite out of date from August 2021. We've moved on. And I want to talk a little bit about just diversification of the virus. As of last summer, when I made this particular slide, there were millions of submissions already to GISAID, the global data sharing of virus sequences, including SARS-CoV-2. We get a mutation around every 2 weeks-ish in the virus, and so we can draw these beautiful trees that highlight how nothing much happened in terms of selection. Early on, these trees go from, time goes out from the center to the edges. And then we started to see selection bringing us alpha with higher transmission. Beta and gamma probably had some immune evasion. Delta had much higher transmission, and now, of course, Omicron. So early on, we had a pretty much fully susceptible population worldwide, and selection was favoring increased transmission, and that gave us alpha and it gave us delta, and you saw that those impacted our pandemic in Canada and the world very severely. We can model our little population in this cartoon way, where we have an infectious person and we just imagine, okay, you're the virus, you're in this infectious person, you're seeing a pool of susceptible individuals, and you're going to have an advantage over your little cousin virus if you can get into those other individuals more and faster. So if you are more transmissible. We don't think there's much direct selection on severity because severe symptoms manifest after infectiousness is over. So it's not, severity isn't really impacting the virus's replication from host to host directly. Then, of course, we immunize our population with vaccines, and now also with Omicron exposure. And that means that a virus that can get into those individuals who are immune now has an advantage. So early on, we had selection favoring transmission. Now we have selection favoring immune escape. And that's what brought us Omicron and its success. Selection began to favor breakthrough infections, and we can look through some of the work of Kovarnet and Jesse and Carmen's work, looking into which mutations are happening in which viruses in which places at what time, and what do those mutations mean. And we know a lot about the biology of the virus. We know about mutations in the receptor binding domain of spike, and what these do at the viral level and also at the population level. Our knowledge of this is partial, of course, but it's non-zero. And we can start to see how selection began to favor increased transmission. And of course, in particular, that impacted with Delta and then with Omicron, which both has increased transmission and ability to evade immunity. There is a framing that has been really influential that you probably will have heard, that either all viruses or this virus will become less severe and more mild over time. This is a paper from Jenny Levine and others at Rustam Antia in Science in 2021 that basically stated this view very articulately and with models. The view is that repeated exposure and repeated infection results in decreasing severity because your cellular immune response builds up and prevents you from having severe disease. And this, of course, is great. I mean, you've got to have a silver lining somewhere. You're going to get infected with COVID lots of times. Great, it gets less severe. Wonderful. So this would place COVID-19 on a trajectory towards a childhood illness that is mild in adults, that then, because it's so mild in adults, becomes comparatively more severe in children compared to adults, not compared to now. But the caveat here is that it can take years or decades to reach this low severity. So this is from their modeling work, the reproductive number, which depends on the serial interval, I should note, on the x-axis, and how fast sterilizing immunity, so that's protection from getting infected, wanes. And they find that in most of this parameter space, it takes a long time, maybe 5 years, less, but only if we really let it rip at R is 6 through our population, which of course we haven't done. If we compare that to evolution, we find that evolution is faster. That decreasing infection fatality ratio in their paper, same paper from Science, 10-year time frame. If we look at COVID in Canada over the last year, we've seen Alpha, we've seen Delta, we've seen Gamma, we've seen Omicron, we've seen major phenotypic changes in this virus, with Alpha and Delta being more severe than their previous, Alpha more severe than original classic COVID, Delta more severe than Alpha. We've seen immune escape in some sense, and then major immune escape with Omicron being less severe. And so this framing that has been very influential in thinking, well, it's going to be okay because it's all going to be mild forever, does not account for evolution. And evolution is faster. This all happened in 1 year compared to a 10-year time frame in the kind of age-driven decrease in severity. So we can use models to think about how that might work and how that might play out. This is a model fit that I did last week to BC's reported cases, which are shown here. And we tried to account for the dramatic reduction in testing by various ways, which I can talk all about, but I will spare you. The key idea is we can understand the tea leaves of the signals from reported cases, extrapolate what that might mean for the number of infections that are circulating, the prevalence, which we would estimate right now to be about 8%, well, maybe 5 to 10% of BC, peaking sort of this week-ish. We think, but we don't know. But we can also think what happens next. So the number of circulating viruses will, of course, give evolution the pool of things on which to act. Variants will emerge faster if the virus population is bigger because it will have more opportunities to generate those mutations that may be favored. That population, in turn, depends on lots of stuff we don't know, right? How long is immunity against infection going to last if you're fully vaccinated, but you had Omicron before you got boosted, or if you got boosted and then you had Omicron, or if you, well, these things. It depends on the efficacy of our vaccines to whatever virus is circulating. Probably BA2 is the same as BA1, but we don't really know that. It depends on the frequency and uptake of vaccination and boosters. It depends on the transmission rate of the circulating variants. So that's the RT, which again, informed by the serial intervals. And that depends on us, on the virus, but also on us and what we do. And of course, it depends on introductions from other places, and it depends on the impact of current diversity. So all of these things, if you just change a couple of them, here I've only changed, I think, the duration of immunity against infection, and in vaccinated people and in boosted people. And you just get, a factor of 5 difference in the circulating virus population. And then, of course, evolution can add to this and change this trajectory. So how does that work? This is a picture. This is from Sally Otto. This is a model, and the circles represent the population divided into whether they're susceptible, exposed, and so on. And we can just sort of think about our population and like how many people are actually susceptible to the virus. Those are both, some of them vaccinated and unvaccinated, because now vaccinated people are also susceptible. And we can think about like, how does selection, if you see this model through a lens of selection, what do you see? You see selection favoring higher transmission, more virus getting these groups to be bigger. And that can happen through increased transmissibility, innate intrinsic transmissibility, which we got for Alpha and Delta and Omicron. It can happen through increased ability to get into that portion of people who don't have COVID, who are immunized or previously infected. So that's immune evasion, which we got with Omicron. We can select for prolonged infectivity, and we'll be selecting for that more strongly as cases decline. So as cases decline, you as the virus have an advantage over your little virus cousins if you can stay in people for longer. both of these things can affect severity and mortality indirectly. Having more virus, having more ability to get into the next host can have an incidental effect on how severe you are in this host. Prolonged infectivity too can be connected to where the virus is. Is it in the upper or lower respiratory tract, for example? So we can use modeling to think about how evolution might act. We can use virus sequence data in the large population context from GISAID to understand what is happening. And I would like to be able to use genomic epidemiology tools to understand what those viruses mean and what these selection pressures are and how they're happening in our communities. And that's what I think this is going to pull this talk together and where I would like this field to go. So, for example, imagine you're in local public health. You see 25 vaccine breakthrough infections in your community. Are they closely related? Are they different from other viruses nearby? Or are they severe? So these answers shape who is at risk, right? If all of these are really closely related and they're a distinct thing, then you have potentially strong transmission among vaccinated individuals in your community. If they're all quite different and they're all sort of spill over from unvaccinated individuals in the same area, then you are less facing a breakthrough, virus that is transmitting in vaccinated people. So these answers shape who is at risk. They shape how much risk they are at, the severity part, and they shape how heritable that risk is from one virus infection to the next. And these things make a huge difference. But in order to know that these 25 breakthrough infections in your community, in order to answer these questions, you need a virus surveillance infrastructure that helps you to answer them. So of course, our worst case scenario is not Omicron. Our worst case scenario is a variant that is like Omicron, but really severe. One that can get into vaccinated people, cause severe disease, and transmit among them. It would be nice to know and know right away if we see the emergence of such a variant. But in order to know that, we need linked data. We need, I think I've got it here. So we need not just the sequences, which are usually shared or more often shared. With these sequences, you can do these sort of larger scale inferences that I haven't talked about. But there's other data that we could be joining and analyzing together with our sequences. And those would answer the questions that I illustrated in the previous slide. Relative transmissibility would come from epidemiological data. How much risk an individual is at would come from clinical data. And how it's playing out with vaccines would, of course, come from immunization data. And if we want to ensure ourselves against the worst case scenarios, we will need not only sequence data and these other data, but links among them, because we will want to be able to answer the question, is this a new virus? Is it infecting vaccinated people? Is it transmitting among them? And is it causing severe disease? So we've written a piece together with David Earn, Nick Ogden, and others from these places that sort of articulate this view. And I think it's under review at the moment. Okay, so I'll draw to an end. Genomic epidemiology can offer some powerful analysis tools, and we hope that our serial intervals and plausible trees will be used, and they're already being used a little bit in Melbourne and hopefully here in BC. They can potentially give a high resolution view of transmission dynamics. But in order to make better use of these data, we need more links and we need more of a picture, more eyes on the data. It's a complex pandemic. The evolution is complex. It's happening quickly, more quickly than our policies can adapt. And if we can put many eyes from the scientific community on these data, we can ensure we learn as much as possible. Okay, I want to thank people here in bold contributed slides, my own team at SFU, collaborators in Melbourne who provided us the data, Sally Otto at UBC, the stuff about data sharing and the kind of vision for, really, we do need to link some of these pieces together. That piece is joint with these folks here, and I'd like to thank my funders. Okay, and thank you all for coming.
Fantastic, thank you, Caroline. Lots to think about and digest. Opinions are my own. I share a lot of those opinions as well. But yeah, I'll open it to questions. So feel free to put them in the chat or just raise your hand and shout out.
Yeah, and let me know if I should stop the share for a more, conversational view, or if people have comments about the slides, then I'll leave it up. Maybe we'll get into it a little and see what the preference is.
Yeah, I see Celia first and then followed by Alex.
Thanks, Carolyn, for a lovely talk, thought-provoking. So, as there's more and more active infections around, then there's also going to be a possibility for somebody to be exposed to multiple different strains of the virus at the same time. Is there any data on that in terms of whether that contributes to susceptibility or severe disease or anything along those lines?
Yeah, I don't know. I suspect that, so the idea you're talking about is co-infection, where you have infection with multiple types at the same time. In Canada, we certainly wouldn't know. We're not testing the first infection, never mind the second one, and we're mostly not sequencing. So we would be flying completely blind to that. I would say, I suspect that from, neutralizing antibodies, you would be protected from that second infection to some extent. So reinfection is probably somewhat rare, but it's much more common with Omicron, or would be much more common with Omicron. So if you have Delta first and then you get Omicron, and Omicron is able to evade the immune response you have from your spike from Delta, then you'll have more common reinfections. I think it would probably still be relatively rare, but it would give the virus opportunities to recombine and sort of mix its best features of different lineages together, which is obviously not what we want. And it does have subtle differences in how much that's possible. Actually have long-term impacts on the modeling and on the population dynamics. Do we expect Delta to still be around, or do we expect Delta to get outcompeted by Omicron? And those are questions that it's hard to answer without that information about cross immunity. So I think there's probably some good protection. There will be some reinfection. It's probably going to be rare, but it'll be less rare if 10% of your population has COVID. And that will be a source of new variants and potentially an ongoing diversity in the virus.
Thank you.
Alex, you can go ahead, and then I see Anthony.
Hi, thanks for the great talk. That was really interesting. So this question is more about, I guess, like a communication approach, because I guess what caught me off guard was the part about the narrative of, COVID is mild now, or COVID is endemic. I see a lot of sort of armchair epidemiology, especially in the media, and I wonder, I don't know if this is in your wheelhouse, but I guess how do we keep the actual sort of scientific stuff at the forefront of these discussions? Because it's, with public health, you have to make sure the public's kind of following along with what you need to do. I know that's a real hairy question, but that's what came to mind for me.
Yeah, I mean, I think if scientists knew that, we would not have the world we have today. Like if we knew how to keep science and the truth at the forefront of discourse, we would imagine what the world would be like. So I agree, it's a big question, but and I don't want to just throw my hands up and say, oh, we don't know that. I think we try hard. We do a lot of work talking to the public through the media. It helped that the WHO came out and said, this is not actually mild. People are actually getting severely ill. I think that did help. I think public health institutions should have a leading role in not falling into the politics of saying, oh, it's okay, it's mild. I think one of the things I've learned in the pandemic is, and maybe other people, or maybe I should have known this before, man, wishful thinking is a powerful force, really, really powerful. And we're all, we want to be wishful. We want to think, oh, cellular immunity is great. But actually, if you crunch numbers, you don't have to have something much more severe than Omicron before you wipe out our protection against hospitalization, because a lot of that came from being protected against transmission. Omicron is, you're half as likely or whatever to get hospitalized. And that's great, but it could be more severe next time. Alpha and Delta were more severe, Omicron was less severe. So yeah, I think it's a great question. I think we have a job to do. We have a role to play as scientists in communicating with the public, trying to do that. We spend a lot of time on it, and we should be thinking about how to do that. And so should our public health institutions. They should be responsible and try not to succumb to wishful thinking, and they should not be as politicized as they are.
Thanks. I don't have a webcam on this machine, but I was nodding along with you the whole time.
Okay.
Great. Anthony, please go ahead.
Hi, Caroline. Can you hear me now?
Yes, I can hear you. Hi, Tony.
Hi. That was a great talk. Thanks very much.
Thanks.
These maximal likelihood phylogenetic trees, which you were then calling plausible trees later on, they always worry me. How stable are they to, like, when you get additional data and recalculate them, do they still look similar? And how much does that matter anyway for the later numbers you calculate?
Yeah, okay, those are great questions. I will make a distinction. So the phylogenies here are, I used them just to illustrate what the population looked like. We don't actually use them for this work as inputs. So sorry. Here's
Okay.
a phylogeny. These things are not particularly stable with really, really closely related sequences. This is more of a way to illustrate that we can use genomes to think about ancestry. When we say plausible trees, we take genetic distances from either a tree like this or from an evolutionary model directly. So we say a sequence 1 and sequence 2, how far apart are you? Do transitions matter or transversions? Or like, what's the genetic distance between these two things, taking into account different rates at different sites and different transitions and transversions and blah, 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 to just get a pairwise distance. Then that pairwise distance we use in this stage. So when I said plausible transmission tree, I meant who infected whom. Did A infect B or did A infect C? So
it's just the local info, which is much more stable.
That's just local info, which is much more stable. We do have a different preprint coming out about the impact of, when you add more data and you reconstruct a tree like this, and you make clusters that are defined on that tree, it is actually very unstable. And that's one reason you probably shouldn't do that and define clusters for public health purposes through a process where you make this maximum likelihood tree, which has all these nearly indistinguishable virus sequences in it, and then you draw circles on that tree. That can be quite unstable, and that is not what we did here, but it's a really important point.
Thank you.
Thanks, Tony.
I'm going to have to run to go teach. So I'm going to hand off moderation duties to Celia, if that's okay. So I'm appalled, but
thank you so much, Jesse.
Yeah, thanks for coming, Jesse.
Yeah.
Talk to you soon.
See you later. So yes, are there any other questions for Caroline? One of the points you had made is that there wasn't an appropriate surveillance going on. So are there other countries that are doing surveillance that would answer some of the questions that you can't get here?
Yeah, so it's a good point. We are doing some virus surveillance. We're doing some sequencing of viruses in Canada and uploading them to the shared databases. It's all changing now as we radically change our testing infrastructure. So I think we do need to design systems going forward that can answer these questions, or we need to think about the purpose of the surveillance system. What's it trying to do and how is it going to do that? The UK has world-leading data in this framework. They do sequencing. They have links between sequences and other data streams, and researchers can get access to those through data sharing agreements with Public Health England. I don't know if they join in with Wales, Scotland, and Ireland, but at least within England, and probably more broadly, they do have some of this infrastructure. I should say it hasn't stopped them from having an epic pandemic. It's not like if you give scientists the data, they go, oh, let's stop this and we'll figure out the policies for you and then make sure they're politically palatable and they happen. So, I think we do need to do these things, and we use UK data a lot. Denmark and the other Scandinavian countries also have really great surveillance. None of them have the provincial jurisdiction in public health and our unique Canadian federated system to contend with. England to some extent, obviously with the 4 UK nations, the broadening beyond England could be challenging. So yeah, there's I think there are surveillance systems that have done those links before, but I'm not sure anyone quite knows what the ongoing SARS-CoV-2 surveillance should look like. I do think in Canada right now, we are kind of flying blind. Like if the new Omicron ++ happens in Canada, we're not going to know soon. Certainly external scientists aren't going to know. And even within public health, if you only have BC sequences, but something is shared from BC and Quebec, you just don't have the statistical power to understand what you're seeing and to raise those flags early enough. So I think it's a big challenge.
Mm-hmm. Other questions? Yep, Carmen. Oh,
Carmen just has to go.
I gotta go, sorry. Bye. Thank
You waved at the wrong
you. It
moment.
was great.
Bye, Carmen. Thanks for coming.
Bye.
I think we're reaching the end. Students in quantitative life sciences, please don't forget to stay on the same Zoom link for your chat with Caroline. And at that point, I will say thank you so much for a really interesting presentation.
Thanks for coming, everyone, and for the great questions.
And I hope to hear from you again sometime soon.
Yeah, well, maybe next time we can meet in person somehow.